Hello and welcome to another episode of Oral Pathology Tuesdays. Today we have the research series, and we also have some very bad news. We just lost last week one of our own colleagues, rather two of our own colleagues, Dr. Shaheen Ahmed, who was the former professor and head of oral pathology at the Tamil Government Dental College and Hospital, passed away, and sadly so did her husband, Mr. Ahmed. And from some very sad news we got, so did his son, Dr. Parvez Akram. Now, this has been sad. There are currently really the loss is more than just these people. I think over 3 million people have died ever since COVID has started around the world. And this seems like a good time to just take a moment and say a prayer and hope that everybody gets past this. Yeah. Now, with that, I'm here with a talk on research. And also with me, I have Dr. Nandini, TB, Professor and Head, Department of Oral Pathology, Dental College Regional Institute of Medical Sciences, Imphal, Manipur. She's going to be moderating the session. We also have Dr. Aman Chaudhary, who is for the first time, in fact, on the channel. He is a professor, Faculty of Dentistry, Jamia Millia Islamia, New Delhi. Yes, and welcome, Dr. Aman, and welcome, Dr. Nandini. Thank you for being here. It's wonderful having you all here. Talking today about finding a research topic. And let me start by asking you a question. How important do you think finding a research topic is to successful research? If you think it's really the main thing, maybe you can put a comment in the chat and we will see what you think about it by the time we finish. And then you can let us know by then what you still think. Is it the main and the only thing that makes a difference? Or other things also count? Really, I think many of you would think that finding the topic of interest is it. I mean, you have the topic and you're done, right? Well, that's not quite so. It can go very wrong. Because if just finding a topic of interest and finding an original topic was the way through getting your research done, then my original research should not have been just over 50, should have been over 500. So I'm always brimming with ideas. But it's not always that easy. Starting 28 years back when I was doing my, when I had to do my own dissertation, within the first 6 months, I could choose my topic. I was definite what I wanted to do. And I know today we expect everyone to be actually submitting their abstract within 6 months, but in those days we had a lot more time. It was like over a year. So keeping that in mind, I was pleased as punch. I had my topic. I was way early. So what could go wrong? But a lot went wrong. I chose a topic. You see, my first seminar was on caries. I fell in love with the topic. I could find an original research, and there I was, all there and set and ready to go. But then I miscalculated. You see, choosing a topic in a setting where actually no one else was in any way really interested in the topic in the department. And there was no particular expertise. So, of course, everyone wanted me to get the famous key article. Now, question is, how do you get a key article for something that's original? You just can't. After a lot of trouble, finally, the microbiology department stepped in and they supported me. And in fact, I think many of you would know Dr. Kishore Bhatt. I was among the first oral microbiology things that he did. He helped me with that study. Then we had technical issues. At that point, it was not possible to get pure culture. You actually had to anything of Streptococcus mutans, least of all. So you had to actually culture the sample. Now, as mutants, as we know, was an accidental discovery by J. Clark, and it seemed like it had grown very easily. I mean, it was accidental. And here I was trying to cultivate it on a selective, differentiating medium, feeding it all the right things, and it was not growing. So, desperate times, desperate measures, all was left was I did not brush for 72 hours. I had an unprecedented amount of snacks and sweets. That part was not bad, but well. And then finally, I could grow the microbe. We did it, and I could go on. Now, I'm really telling you all this not really to grumble or in any way say that that was bad or it was an experience that I would have done it any differently. No, I would have still done it exactly the same way, given the chance, simply because I thoroughly enjoyed it. But then it also made me realize that just just the topic is not enough. Just an idea is not enough. What we need is, like, I spent years now to figure out how, but finally I've come up with this. There's a way to doing research, right? It's like every definition we have in dentistry. It's an art and science of the perfect research topic. And how that goes is to understand first a lot of things about identifying the topic and a lot of things about everything else that goes around it. It's not just the topic. So let's just look at some things. And let's start with some reality check. The very first one is that eureka moments are like pure fantasy. And lovely as they are, as fantasy is normally, but they are still nonetheless a fantasy. We have all seen this picture of Archimedes supposedly in his bath and finding out everything about the laws of displacement. But the thing is, Archimedes was not just another Joe getting in a bath and then finding about displacement. He was already a scientist of great renown. He had been working on both laws of movement and on water because he had made the first well. He had done a whole lot of things, and and at that exact point, he was actually trying to figure out how to check the purity of a gold crown that King Heron had given him, and obviously without damaging it. So when he got into that bath, this whole problem was on his mind. And keeping in mind that he was already an expert in it, well, he could come up with it, sort of fell in place. He could come up with an idea. And just one more example, of course. This is an image again, all of us are very, very familiar with the very famous one of Sir Isaac Newton sitting under the apple tree and finding out all about gravity just because an apple fell. Now, the fact is, had he not been Isaac Newton, had he not known so much of physics already, he could have sat there and been, showered with apples. He could have sat there until an apple was no longer just an apple, but was a phone. But still, he would not have come up with the thought of and the laws of gravity. What happened was that, Isaac Newton was already a genius. He had a profound knowledge of physics. He was obviously obsessed with it. I mean, he was sitting in a garden under an apple tree in beautiful scenery, and he was thinking physics. And then he had a good power of observation. So when he saw that apple falling, it really just made everything fall in place. And that's it, nothing more. That apple did not have magical powers. So what does that mean? To find a new topic, you need to be a genius? Well, and not so, not necessary. The profound knowledge is essential, definitely. And obsession is good. They say almost every researcher should be obsessed about what they're researching in a healthy way, if there is something like healthy obsession. But anyway, focused interest also will do. Maybe you don't really need to be obsessed. And definitely you need good power of observation. And if you have all of that, really finding a new and novel topic is not a big deal. But now about that a little bit later. Let's first start with what is research and what is the research process in very short, because this is really not the main heart of the talk, as I know. So to put it very simply, this is a very, I know it's like a busy slide or a picture that I had made some years back. So research is about developing a hypothesis or a theory to answer a question which is aimed at identifying or explaining a previously unknown fact. So basically, you could be either identifying something totally new, or you could just be finding something a little more about something that's already known, something at the peripheral, a little more about it. The whole thing is done through a process of research. Of course, literature research is already essential in the background search. So, all the background that we need, all that in depth knowledge that you need to come up with the hypothesis is definitely there. Then, also, research, the whole process itself can be a literature research, like a systematic review. Then you could also be doing it, which is what happens mostly in what we do in terms of doing a master's dissertation or a PhD dissertation, or most of the research that's done in the departments, is either experimental or observation. Now, the very heart of research says you should be able to have a predetermined study design. You cannot sort of wing it as you go. You have to have predefined and quantified parameters that you're testing. So, again, something that you cannot really wing it and modify it as you go. And at the end, the whole thing should be appropriate and unbiased when it comes to analysis of results. And you should also be able to report the results, whether they are positive, negative, or in fact, find nothing. That's a nil output. And to cover all of this needs to be repeatable and reproducible so other researchers can actually do it and see if everything was right and build up on that, because our knowledge is essentially incrementally building along this. And again, the whole thing needs to be ethical and unbiased. That's a lot. Now, to put the whole picture, when you think about the research process, the first is, of course, our question or the hypothesis. Then we come with the design. Then the statistical methods have to be also found. Any permissions and funding has to be decided. And only at that point, you are actually ready to submit a proposal or a synopsis, as it may be called. And then the study itself goes on. But simultaneously with this, for the picture to be complete and for it to actually be a hypothesis that can work, you need to also, side by side, literally at the same time, start and find out certain other things. Besides the fact that you need to still, and I keep emphasizing this, find one topic that you are going to get that profound knowledge about. Then you need to very fast and very early find out what are the dates for submissions, what are the formats, what are other requirements that may be there in your institute or in your university. You need simultaneously an understanding of bioethics. What are the types of study? Because that will eventually go into your design. The levels of evidence, which is again something that will go into your design, biostatistics, and all the various available resources. Now, if you do not judge all of these together right in the beginning, you could end up having problems like I had, and it could take you a lot longer. It's not impossible, but it will take longer. And yeah, there's something that I have always wondered why it sort of is not so clear to us. This SMART goals, or goals and objectives that are SMART, is a whole acronym that is a very business originated one. And because businesses need to have results and outcomes a lot more seriously, and somehow maybe on the science side, we don't take it as seriously, but it is required. So what is a SMART goal? Basically, you need to see that what you're looking for is specific. You cannot be researching something too vast and too inspecific. Like, for example, you can't say, I'm going to check every microbial species in adult mouths of this population. It's not specific. And it's not possible. Because it's not specific, it's not possible. Then the second thing is it should be measurable. Now, measurable is in two ways. One is it should actually be possible to measure. And then it should be, sort of in existence to measure. So, for example, again, going back to a microbial example, you cannot say, I'm going to count all the bacteria in someone's mouth. We know it's not possible. We don't really actually count. It's like attempting to count the sands on certain beach. You can't really count it, not grain by grain. You may be able to estimate it, but you can't count it. So your measurement and measurable ways have to be correct. Another thing is, say, for example, your salivary pH. You can check someone's salivary pH now. You cannot by in any way figure out what was someone's salivary pH before. Salivary pH, blood pressure. You can make some assumptions based on some other factors, but you can't actually measure it for a fact. And the next thing, of course, it should be achievable, both in terms of there should be technology that makes something achievable, possible right now. You could also have other various restrictions, like, for example, we have restrictions today on traveling. Sorry. So while years ago, I could say that I'm going to find the distribution of a certain disease or the way a certain disease appears in, say, in the state of Karnataka or in South India. Today, I can't because obviously I can't really travel to those places. And if it is something that requires a physical examination, not possible. So with times, with situations, and with your current status, it should be achievable. Then it should be relevant. I think the best example I can say, there's no relevance, for example, today for anybody to try and study something like smallpox. It was eradicated long ago. What would be the point of studying it, right? So probably if someone is studying it, it's not for good reasons. So anyway, and that we are not interested in. That's the thing. It should be relevant. It should be relevant to the population it is being studied on. That's another thing. If a disease almost never happens in a population, why study it? So then it comes to time bound. Well, we know that we need to submit your dissertation. You have to finish the dissertation. You should be able to finish it in that certain amount of time. So, for example, you can't really start, today on a dental caries study, which we know sometimes, most likely, the average time we take for a cavitation to occur is somewhere anywhere between 6 months to 2 years. Now, in our 1 year practical time that we get for our dissertation work in a postgraduate in the master's degree is obviously not enough time. You can't hope that every one of them is going to develop that cavity in 6 months. So it should be time bound and it should be time relevant. You should be able to finish it in the amount of time that you have. And these are smart goals, which I think are really essential to the success of any dissertation, any thesis, any research that you do. Now coming to the actual work. The very first thing is to find a really good topic, one topic that you really are interested in, and become an expert in it. Now, the whole thing of, I think on an average, and I'm talking mostly about our master degree students, masters. So you may have about 6 months before you have to submit your synopsis. So clearly you have to have your question within the 6 months, well within the 6 months. So you should have your topic that you are really good at and be good at one topic. And I would say that should not take more than 50% of whatever time you have for your synopsis or proposal submission. You need to build that definitely within that much time. Now, it may be much easier said than done to say, go find the topic and become an expert in it. How do you choose the topic? It's easier for PhD students because they've already done their masters. They already know quite a bit about their specialty. So there is no problem there. But what about everybody else? It's like everything else. You just manage it. You just work through it. There's a system. You can go through it and you just go step by step. So the first step and the first thing to remember is within the biomedical areas of biomedical systems, almost everything is focused as two things. One is state of health, one is state of disease. So everything that is health, we would like to define it, we would like to quantify it, and then essentially we would like to maintain it. While everything that is disease, we would like to be able to diagnose it. We would like to know what is its disease cycle, what happens if it's left untreated, what happens if it's treated. We would like to know its distribution, so epidemiology. We would like to know management, which of course includes right from prevention to treatment and prognosis. And every bit of technology and every bit of every process, every technology, every system that supports any portion of these things is all game, is all open. to study for us. There's literally the sky is the limit. If you can identify within these what you like, it makes a lot of difference. And it could, and essentially, since almost everything in the world can relate back to us, and we are human beings are rather self-centered, so the whole world sort of rotates around us, and that's the way it is. So almost anything can be brought back to this. For example, let's say you love music, you have some natural talent in music. If you want to study music with any of these, you can. You just have to find the right question and combine them, and then you are within one area of your strength already. Also, if you have a natural love, now I had one of my students, she had joined oral pathology, but she loved oral surgery, or rather, she loved oral pathology, but also loved spending time in oral surgery. So we already knew this. And then when we were trying to find her a topic, we could give her one that required her to spend a lot of time in oral surgery. And of course, obviously, come back to oral pathology and do work. And she did a very interesting, very original study that had great application, in fact. She checked the intraoperative uh, imprint evaluation of the surgical margins in oral squamous cell carcinoma. Now, this was a study that actually was used because we could replace something as expensive as frozen sections. So, which the technology is so much more expensive. And at that time in courts, and in fact, even now, the Department of Oral Surgery has an in-house OT, and they do a lot of oncopathology. So this was a great work. This was a great piece of work. Both departments really benefited. We worked together, and we could only do this. And she was delighted doing it, and she did excellent work. And we could only do this because we knew that that was what she loved, and we just combined some things together and gave her a topic that she really excelled at. Now, besides that general thing, so how do you really narrow down to the actual exact topic? Now, of course, I'm going to give an example of oral pathology, but frankly, this can work in any subject in any specialty. So you take your main specialty, the main heart of it. But around that, we inevitably have different disciplines. Now, within oral pathology, we know we have pathology itself, we have microbiology, we have forensic, we have laboratory techniques, we could have genetics and other things. Now, from those, you choose which one. Which one you like more? I think this should be fairly easy to know what you really like more at this level of distribution. It is quite easy to achieve that. Now, once you do that, let's say you find that you like pathology. So, and this will go also obviously applied to any other one of those disciplines. If you can, you will just have to, it's a process, you just have to follow the process. So from there, you go to disease groups. Now, the next level after the initial disciplines, if you just pick up any of the average textbooks that we have, so they're all, you just go through the content page, you don't need to do more than that. The diseases or disease groups are essentially divided either in disease groups. Sometimes things are divided by systems. So you could have, diseases of skin, or and of course, if you are in if you're not in a pathology department, it could be different fields. I guess if you're a prosthodontist, you would have, fixed prostheses and removable prostheses and stuff like that. Anyway, so you choose. Now, this is where we are. And of course, that list is not complete, but just an example. Then what? You look, and it's probably not that easy to choose again. What I have found is if what you love doesn't sort of hit you in the face, the next thing is to start deciding what you definitely don't want to do and start excluding those from the list. And once you do that, maybe you have one or two, and then you start again thinking, what should I do with it? Let's say we chose dental diseases, and then we have now individual conditions. So individual conditions, and of course, since it is me giving this example, I decided that I would leave caries behind. But this can be anything. So now with this, we have one topic, one area, it's not really that daunting to actually reach a level of great amount of information, great amount of skill and knowledge in one topic. And remember, you have half the time to finish doing this. Now, this is, you have just identified the topic in 2 to 3 weeks. This is sort of, it should have taken you 2 to 3 weeks to achieve this. You have identified your topic. Now, next, you need to really concentrate on that one topic and find out more and more and more about that. And how you do that is first, really, by just studying everything that is within the current textbooks, and then looking for some review articles. And as you do that, the easiest way is to make topic maps. So, you know, if you have caries in the center, you have what all about it, you have the microbiology, you have the role of saliva, you have, say, pathogenesis, and you have prevention, you have diagnosis. So this is all what, and relating again to what you're doing. So if I guess if you are a conservative dentist, you will also have a lot on treatment in there. And you choose your topic and you start building on that. That's, I think, quite simple actually. But simultaneously now, that those 2 to 3 weeks were not really just time off for you to choose a topic. It was also to do other things which are equally important. So at the same time, you need to first and foremost find out what are the formalities required. Finally, remember why you're doing all this is to be able to first submit your synopsis and then eventually do the study. So what are the formalities for submitting that synopsis? What do you need to add there? What are the forms? How many copies? You know, different places have different formalities. Who needs to sign it? All sorts of things. And you can find that from your seniors, you can find that from your teachers, and of course from the office, administrative office. And the dates, very important, the dates. Please put these somewhere very noticeably that you will see every single morning. What is your date of submission in the department? What is the time? What is a likely date where you will have everything has to go to the IRB? So when is it going to have to do that? And when is the final submission? And from my years as a teacher, I can have this one little note of advice. The date of final submission is not the day you decide the topic. They are not the same thing. I think many teachers has probably felt like pulling out their hair when they realize that on the day of submission or the day before the submission, students are just telling them, okay, now I found my topic. And that doesn't work like that. You need a lot of time between choosing the topic and actually preparing it in a study-worthy manner. So keep that in mind. I keep it noticeably around you. At the same time, bioethics is something you need to study in case you are doing anything about studying on humans, human tissue as well. You need to know about bioethics. So this is a good time to begin to study it, to begin to learn it. There's no point in waiting. And really the first few weeks of your course are rather slightly free and not much is happening. It's the best time. You find them online courses. The US department has them. I think ICMR has them. Probably most universities have them. And then there are, of course, loads of books and online courses. Just get it done. There's no point in waiting. It's something you need to do. Might as well do it. It will help you in preparing your study. Then, not to go too deep in it, but although you do need to study this quite in depth, but here is just to tell you what these are now. Levels of evidence. You will need them for designing your own study, and you will need them to be able to understand what others have done and what you can use, and if you need to take one of those studies to the next level so that that becomes your study. So this is something you need to familiarize yourself with in a very simple and basic way. If I had to say, starting from the lowest is a case report. Now, a case report is exactly what it says. It is one person's report. It could be even your own case, but you find something interesting, something rare, and it is obviously not good enough. It's not something you're going to do as a research process, but it is, these are the details sometimes on which you can begin to format a hypothesis. If you have come across something interesting, maybe you can form your hypothesis based on some interesting thing and then see, does it happen? Does it not happen? So case reports are at the lowermost. Uh, it's like we had this very interesting thing, one study we were doing in oral submucous fibrosis, and one of the patients, an 80-year-old person who for the last 60 years had been smoking and chewing tobacco, had oral submucous fibrosis, and he very clearly said, and he said it to first one of my juniors, checked. Then I said, no, you must have made a mistake. So I went and talked to him myself, because of course there is strong evidence that, and we keep talking about how areca nut is the only thing that can cause oral submucous fibrosis, so much so that we don't even look for other things anymore. But so there was this gentleman, I talked to him myself, and he said, listen, I'm telling you I've smoked for 60 years. I'm telling you I have been chewing tobacco. Why wouldn't I tell you if I was taking areca nut? What difference it makes? And I know I'm not taking areca nut, not even rarely, because I don't like areca nut. And he was very specific. So well, I can say that's interesting. And I can definitely go around looking, you use that as a basis for another study, but it is not sufficient proof. Then coming to the next level is case series. So case series is basically you had one, now you have a whole lot that are showing the same thing, and you have a case series. Again, rather low on the rung. And of course, case series do not become research. Not the kind of research that we are talking about. The next level are cross-sectional studies. They're exactly what they say. You look at one population at one point in time. Cross-sectional studies. And these are also low in rung. They are good for identifying relation. They can never identify causation. So basically you have two things, three things, and you say they relate to each other. They are coexisting with each other. But the point is A and B may coexist. But does A cause B? Does B cause A? Or in fact, are both A and B caused or modified by a C that we don't know about? Now, this is a shortcoming of a cross-sectional study, but it's very good. It's cheap. It's quick for a beginning. When you have a novel, a new idea, it's an excellent way to check it out, because you don't want to go and spend a lot of things on something that you have no immediate proof on. So that's what cross-sectional studies are for. Then the next level is case control studies. To put it very short, this is case control studies look in the past, and they look at a disease, the outcome of interest, which is the disease. So you have a group of people, and it's really good for diseases that are rare. So you're not going to find many if you go looking for them. It's simpler if you actually just identify people who have the disease. So case control studies do that. You find people who have, say, any disease. Let's say they have a rare tumor, and then you go back in time. You ask them through their history to what all have been they exposed to, what all have they done, what has been their lifestyle that could have brought, that could correlate to the disease, right? And that is a case control study. A cohorts are prospective studies. They are basically, the idea is they're observational. You're observing people. You are not doing anything. But in cohorts, we are actually looking at causation, or rather, not at the disease, but at what could have caused it, at the relation. So, for example, what was the factor of interest? Let's say now after this whole period ends, we could have a group of people who were exposed or who turned positive for corona, and we could have a group of people who did not. And we follow them in the long term to see what possible effects, what are the possible differences are there in their health that could be related to this. Now, obviously, as I'm saying it, you can make out that cohort studies are long-term, they're prospective, so they're rather expensive to conduct, and they're obviously observational. So you're not interfering with anything, you're just watching them. but they have a very good level of, they are essentially a very good level of evidence. And RCTs are the final ones. RCTs are interventional studies. It is mostly to check for some treatment, some procedure, some test, and these are normally never the first thing on the rung, because obviously they are on human beings. And these come a little later on when you have some other basic study that supports your finding. And then the next thing you can do is you can do a randomized clinical trial. I don't think randomized clinical trials probably, they can be done in short term, but in this, again, you should be, then it is not the very first study. It should be something you are following up someone else's study. Uh, they are very stringent. You have to do them really very well. And you have to follow all the rules and regulations. You have to do everything very well, but these are even more so. On the top of the ladder are meta-analysis and systematic reviews. So basically, this is you're collecting data from everywhere, putting it together. Either if it's a systematic review, you're just basically taking something and coming up with an explanation to say, through the years, this is what has worked out. This is the current picture we have of something. Meta-analysis with the word analysis. So you're basically going to be applying all the previous results to analysis and find out where we stand. Very good. And the final word, essentially, as far as level of evidence goes. But again, doesn't really, I don't think any of us is going to do a meta-analysis for a research topic as far as a dissertation goes. But this is something you need to definitely know. This was seriously shot through it, but you need to definitely find out more. And these are things you need to learn at this point. The next thing you need to know are the types of study. You have primary, secondary, observational. Primary is, of course, study that you are doing. Secondary is from the literature. Observational and experimental, both of them can be either on humans or animals or benchtop. Benchtop is benchtop, like where we do we do an entirely lab test, tissue culture, or you test the mechanical strength of something. So that's benchtop. And epidemiological, we are looking for distribution of either a condition or an exposure of interest at the population level. And this is again something very interesting for those who would like to, but you should have access to that many people. Now, to essentially ensure that you don't find yourself in a situation like I did, you need to also have these other things. And one of them, you need to find out, and why I'm telling you to do this now, essentially, and not right in the beginning, is because by this point, you already know what is the topic that you're interested in. If you go and speak to experts in your department, whether your own boss, your own guide, your teachers, very often you may get very, very influenced by something they like. But that kind of influence and that kind of what should I say, liking that is by diffusion is not permanent and is not strong enough to carry you through the whole ups and downs of a research. So what you need to do is you need to first identify what you like, and then talk to the experts around, whether from your own department, in any other departments of the same institute, or even in any other institutes around. And with today's world, you don't even have to limit yourself there. You can get on ResearchGate, find out people who are working in the same field. You can get on LinkedIn, again, establish connections with people who are in the same field. And all of this is very interesting and very important for you to know, because when you get caught up with some way or something, you want to do something, you don't know how, these are the people who are going to have to step in and help you through it. Technology, of course. You need to know what's available in your own department, in the entire institute, and definitely within reachable distances. What can you access? Now, if you're in some very small town at the other end of the country, and you want to say access, here, like I said, Dr. Butt is one of the people who does a lot on oral microbiology and advanced techniques. You may find it difficult to get him the samples in the right amount of time. So you need to know what technology is available where, and you need to make the best of that. Infrastructure. Say, for example, you want to do an animal study. You need to have an animal house close by. in access. Otherwise, it's best not to look at an animal study. Stuff like that. So these are the things we need to know. Samples. What sort of samples are available to you? Do you have a department where with a huge archive? That helps if you're going to do an archival study. But there is a little query there because sometimes even if you have a lot of material, it's good to go back and see how many of your predecessors have done the same thing, especially if that material can get exhausted. For example, our tissue blocks. We may have a lot of them, but if everybody is going to study the same thing over the years, they're going to be eventually empty wax pieces left behind with no tissue. So don't fall for that. Also check up and see how much of material is actually there. Sometimes it may be better to look for something a little less commonly done if you're looking at your own archives. Of course, there are archives which don't run out. For example, if you're looking at images, they don't really run out, they're going to be there. So that's a very different situation. And again, samples, if you are thinking about patients, look around, hospitals around you, see what are the diseases that are common, establish your connections, and go and see if those people are ready to help you. There's a lot of information, there's a lot of interaction required if you're actually going to meet people. That just because there's a certain hospital with a super specialty hospital treating people of a certain disease does not necessarily mean that they're going to give you access to their patients, because there's a lot of different ethical issues involved, permissions, clearances. So go and meet those people before you really get ahead with yourself on those. And funding, always a big deal. I know today we are, at least in India, finally we are getting some funding probably from ICMR for research, and maybe the universities are funding. But still, you have to find out, if you don't get the funding, can you do it? If you're going to think of something very, very expensive, then that is going to be a problem, and you have to already be thinking of where you're going to get the funds for it. So these are all things that you need to know. Now, sorry. Okay, now coming to choosing the topic itself. We are almost there. So what is the best way of choosing a topic? Now you have all this information. Think of there's this whole concept that is called adjacent possible. So you're basically looking at what is already known, and at the periphery of what is known, you're going to look for something that is slightly new or unknown. It was a concept that was popularized by Stuart Kaufman. And I think we have been doing it long before this without really giving it a nice fancy name. So, that's why all your teachers tell you, go to the library, read. What's all that reading supposed to do? Is basically for you to find out what is known and then to begin to look at the periphery of what is known. To give you an example, now, I was at one point trying to do a study on oral submucous fibrosis. Why did I think of oral submucous fibrosis? Because I was having this newfound interest, or rather the need to work on oral cancer. But more than oral cancer, I found oral submucous fibrosis a lot more sad, I think, in a way, because they really suffer, but nobody pays much attention to the submucous fibrosis itself unless it becomes a cancer. So I felt there should be something that can be done there. So I combined some of the things that were known. Now, what were the things that were known? We already knew at that point that oral submucous fibrosis is a chronic fibrosing disease. We knew it's habit related. However, the habit does not equate disease. I think the sense that not everybody who has the habit gets the disease. So when you combine this, means there is something obviously different between people. There's, of course, already existing explanations of genetics and nutrition and various things that attempt to explain this. I just thought I will look at it from a different way. And the next thing we knew was areca nut seemed to be the most definitely expected cause and most related. Now, about areca nut, we know it's highly alkaline with a pH that is greater than 10. Then also at the same time, we knew that the fibroblasts in oral submucous fibrosis, they undergo phenotypic change from their typically fetal phenotype, which is normal in the oral cavity. They go on to more the myofibroblasts or the fibrous producing type. So what destroys the original set? What changes the original set was my question. And somewhere at the back of this, I also remember that alkaline exposure causes fibrosis burns in skins. So if your skin is exposed to alkali, you get burns, and those burns heal by fibrosis. And of course, from my days of dental caries, I remember salivary buffering, very important. And it does not have the ability to buffer for alkaline. It does not buffer alkali back to neutral. It can only buffer acid back to neutral, because I guess our body never really thought that we would be putting something that alkaline and foul tasting in our mouth. So, because most alkalis are actually poisons. So, our body has not prepared for it. Now that we go and put in this alkali, could it have a difference? Could that buffering action make a difference? So, from all these known factors, the periphery of it, I combined it and the unknown. So, finding out, can salivary pH and buffering, or the amount of time the buffering took, play a role in causing oral submucous fibrosis? But our finding was that there is a definite, a significant correlation between the amount of time, the return of the salivary pH after each time a person chews. There's a definite correlation between how much time that takes and whether a person has oral submucous fibrosis, or people who had the habit but did not have oral submucous fibrosis. This was a very early study and, of course, needs further proof, but this was something we did. And it's exactly in the way I said. You have the known, the periphery, you combine it, you get the unknown. This may be okay. There was some amount of original thought in this, but I think anyone who knew all of it would have thought about it. So, right. There is this, how did we do this? Now, I had the idea, but what about building it into a study? So first and foremost, we have to decide what level of evidence we want to. Now, the interesting part is when you have a novel idea, when it's not been done before, you essentially can test it anywhere, because after all, it's not been tested, right? Anything can do. So what we did, but clearly meta-analysis systematic reviews were out, because obviously it's not been studied before. Our randomized clinical trials are interventionals. We are not planning to do anything intervention. Cohorts, far too expensive for an original first-time study where you just want to know, is it true? Is there any relation? Case control, where we are not looking at the disease, we are looking at cause, so it was not a case control kind of a study. And case series and case reports were out, but what remained was cross-sectional. And the best way to do something like this, to establish your theory, to identify whether a theory even is valid or not. So what about the type of study? Obviously, it had to be primary, and it was observational, but human. I mean, I can't get animals to chew, and then check, to chew for 2 minutes and then check the pH. And it was at this point not benchtop. It could have been experimental, in which case it would have to be the benchtop, which means if I was actually experimenting, say, on the cells in a lab, if I had done a culture of the fibroblasts, and that is in fact something now someone can do. And the next thing was epidemiological. Well, it was not really possible to go ahead and look for something like this in the large scale. And again, not warranted before we actually know that there is some association. The statistics were very simple. We had to, this is something you would have to do always. One is, of course, you test a null hypothesis by checking the p-value, by really saying whether, if you have a significant relation, you're saying that the results are not by chance, or mostly not by chance. The next thing is, of course, to ensure that whatever results you got fall within the limits of the overall population, and that you achieve by doing following the confidence intervals. And it is a cross-sectional study, so essentially you have to do a relation test and ratios. There's nothing else to do with it. So these all, and this is how you can make out that if you have an idea of what you're doing now, after you have chosen the topic, obviously the next step sort of falls in place by itself, and you just can go through the steps. Now, let's say didn't have those and you didn't have that original idea. What do? Do you give up? Well, not really. You just pick up. This is where this is a true adjacent person. You pick up a study. In this case, let's just pick up the same study. So what we know is that this study had found a positive association between a post-chew salivary pH, the recovery time, and OSF. So how much longer it took, the chances of OSF were more. This part was known. I knew the study design, it was cross-sectional. The level of evidence, again, it was cross-sectional. And we knew the sample type, it was salivary samples, and we knew the findings. Now, if you know all of this about any study, you can go the next level and you can find something new. So what you can do, for any study, this is possible. You can either improve the study design. If there is a technical shortcoming, any shortcoming in the study, whether it's technical, whether it is numbers, and it's going to give you a better finding, you can establish that. So you can do a technological improvement on what was done before. For example, now, one thing we had forgotten, or rather initially it was the design did not work in a way that I was thinking. So we had not done salivary flow rate estimation, which we should have done, because we are talking about recovery time, right? How fast does the pH recover? So salivary flow is very important. And the next thing was that because we ended up having to go out and we could not get these patients being an oral pathology department, there was a problem. We couldn't get patients in the department. So we conducted camps, we went out, and because we were out and almost all these patients were checked outside, we did not do all of them through a pH meter. Many were tested by just a pH paper. So obviously a pH meter test would be a lot better. And I think, in fact, that you can also, the next thing, the best next thing is go to the higher level of evidence. And one is very clearly now is to do a cohort study, association, long-term cohort study, pick up people who have the habit and who have the exposure and who don't have the exposure, see what is the change in their salivary pH through time and see what happens. The next thing, of course, is you take an advanced technique, a benchtop technique, where you go an advanced technique where, like I was saying, you use tissue, you cell culture, and you do this on a cell culture. You test the effect of alkaline or even areca nut extracts on the cell culture and then see what happens. So these are all very simple, actually, very straightforward, very simple. And voila, you have your topic. You have also found out everything that was required to actually get it now in space and in the right format. I do realize that this is stopping at just the topic, but then that was the topic of the day. And if I attempt to go on to the rest of it, I will need not just 1 hour talk, I will need a lecture series. So maybe the next time, and we can go there. So always remember, finally, everybody will look at whatever the results from their own point of view. And this, I think, is a very good example, which I found on Pinterest. And it is true. So, however, even great minds will think of things in their own terms. And it's a good thing. It's not always bad. Just be open to other thoughts. And some useful tips to sort of say bye. And first and foremost, what you also need to do is learn to write a research journal. Every single day, write down everything that relates to your research. Keep a record of what conversation you had with who, what you read, what you spoke about. It makes a lot of different things. It makes it a lot easier. I wish I had done it. I hadn't. This is something I thought of and learned later. And definitely keep a record of everything. And above all, be organized. Organization makes it all much easier. And yet, let all your new adventures and your new research begin. I do hope this was interesting. I thank you for listening and for being here. Now, let's get everybody else inside and have some discussion on this. So I shall stop the share. Hello.
Thank you so much, Matt. Thank you so much. That was indeed a very elaborate, very crisp, comprehensive way of telling everybody how to go ahead. It was very nice, ma'am. As of now, in the YouTube, we don't have any questions. So I request Dr. Raman to start with the discussion.
Yeah.
I think Madam has touched everything. She has opened a Pandora's box, actually, because after this, they are going to be searching for the topic, and that is going to open everything, actually, because that's the beginning of that is the tip of iceberg where everything starts. Though it is the most important part, but then it's, once you start exploring the literature, it really, starts your brain to really look for something. And everybody is looking for something. Ma'am highlighted rightly that everybody is looking for something eureka moment, which is not correct, actually. To disapprove a thing, they feel everything should be significant what they discover. I don't think it should be more that the aim should be a gap, rather than a discovery. I feel. I mean, it should not be that, you will be becoming a Einstein in a day or so. But to follow a process, which Madam has told, is the most important thing rather than discovering anything. Because maybe in life, you might go up and come up with something really great. But if your process right at the beginning was wrong, the product will be maybe right or wrong, but you will not get a feel or passion of what exactly had to happen in a correct way. Ma'am, you can correct me, ma'am, if I am.
No, it's very right. It's very, very right. Very right. Yes.
Because I could make out from your thing that whole the process, whether we are a postgraduate guide, whether we are a student, the aim is to derive something which is actually polluting science. I feel it should be more towards following a correct process. What you get is like, it's a byproduct. You cannot know what. Had you known what was there, you wouldn't have attempted it. That was exactly what was the aim of the entire thing which you said. I feel that way. I'm looking for good questions as such. Maybe offline, I have seen very good questions coming up later on. Today is a day where I can see people. Many of them have logged in, and I think they're spellbound with the process itself, I guess.
Yeah, well, I hope so, and I hope that I think it's something that's going to take some time for everyone to process. Like I said, I have been thinking about this for years to see it's not just one thing. I really have. It's not just one thing. The topic is just literally the tip of the iceberg. It is necessary to have a good topic, but everything else needs to fall in place. And from my years as a teacher, I have found that students, the main thing is they get so focused on very haphazardly looking for the topic and not methodically looking, like you are reading a little here, a little there, and confusing themselves totally. And at the same time, just leaving everything else. It's like I said, my biggest, and I have always loved my students. And yet there have been days when I have had to remind myself how much I love my students, because they would come in front of me and, it's like 3 days to submission. And they're just like, wow, we found a topic. I'm saying, like, what? You found a topic is not the answer. You have 3 days. So, it's a lot of things, and it all goes together, and you can't leave any of it for the last minute. It is generally the message I wanted to give, and I hope I managed.
Surely,
ma'am. Can I add something?
Oh, yes, please.
Ma'am, you are very rightly you said about the different branches where we can opt for the research, like disease and everything, like forensic, microbiology and on laboratory techniques. Along with that, I feel like even physiology, the normal physiology is also, we normally don't do that. Ma'am, like normal physiology, for example, in postmenopausal and premenopausal women, what is the predentine thickness and so on, which was one of the topics of my post-graduation. So even that area is lacking. The research in that area is lacking. So, I mean, you can figure it out where the untouched topics. Most of the topics in oral pathology, they deal with only cancer, pre-cancer, that is a maximum. But caries, for example, caries is the least taken up. And we in CORDS, we had a very nice way of handling, giving each topic, like forensic for one, microbiology for one, PG, and that was how we read about each other's topic, ma'am. So I really feel that area has to be explored. Few unexplored areas, na? So
definitely,
what is your opinion? Dr. Aman is very much into forensic. He might give some inputs as well.
Yes, yes, definitely, yes.
I feel the quest of actually changing a population is very much prevalent in our society, in our research work, which is happening quite often. I will not say that it is good or bad, but the thing is, you should look for a methodology breakthrough also, because anything which is being attempted in, like I see anthropological studies happening, blindly being copied will be wrong, because they are 6 feet. They are 6 feet. The setup which is there might be different. The temperature might be different. Attempting the same thing and comparing those results, or coming up with the same kind, mixing up the data or comparing on the same terms, will give a wrong inference. So just by topic only, we decide that it appears that your research might be continuously wrong in future. So you have to keep in mind a lot of things when you are deciding a topic itself, that you don't end up comparing mangoes with apples and apples with oranges. It's very important that you know the research design which is happening. You know what kind of a registration process is there. If you're going for a Prospero, how to register, whether it is going for a or a cohort study, what kind of registration is required. And that's where guides need to be very sensitized, because once the process has really moved ahead, going back will be very difficult for a student to go ahead, and not involving a statistician right at the beginning itself might be a defeat. When to call it a pilot attempt, when to call it a regular study, how was the sample calculated. I'm not saying that right at the synopsis when you're writing, right at the research topic when you're aiming, you should have that entire setup in your mind. And that's where guide should really step in, that he has a vision when he's taking a topic, that it is feasibility. A is availability, B is we used to say in our post-graduation, the most good qualities of a student is A is availability, A. B is behavior, C is compatibility, or the compliance which he has. D is discoverability. He's himself discoverable and he discovers something. And E is enthusiasm. He has enthusiasm. He's not dull for everything. We had a whole alphabet which our seniors used to make us mug up, that these qualities are required in you. Have you got even out of these 27 alphabets any 5-7? Then you are eligible to really go ahead with your studies. And that's what exactly I could see your synonyms there, those acronyms. Those are very touchy, because they really remind you of what exactly should go around, how you should go ahead, and you might not commit mistake even.
Very true. I think it's really the main thing is just start working on everything from day one, and start working with a collective, a holistic view. Nothing is, because you're talking about research. It's literally, finally, I think a good research is, in my mind, is like a good symphony. It needs to be conducted.
Exactly.
And every part needs to play its part. You can't imagine, you can't think of a symphony if, one whole group or one whole section is playing something entirely different. What you will have is cacophony, you will not have a symphony. So, which is what happens. And I think it's very important. What happens, I think, with most students is that they realize this a little too late. When they're already almost at the end of the time allotted, they realize that, oh, I should have done this before. So the idea is we have all those things And they know from day one, I need to start. 6 months is not a long time. That's why I said, keep that date noticeably somewhere that you can see it. 6 months just off like that. Before you know it, it's done. And there's so much to know. And it all needs to literally merge together. At the end, that topic you have found only makes sense if you also see getting it in the right study design, using the right statistics, you have the funding, you have the samples, you have the technology. You have to have everything before you can actually make it work. Otherwise, it just doesn't work. So it makes it's something that just needs to come together.
One more thing is, any researcher, when he does his research, he should put in what difficulties he faced and what were the loopholes in their research. Not the loopholes, in the sense the drawbacks or the difficulties they faced in, so that the next researcher who can carry on with those things, not repeat the same study. So that is one more thing that is needed.
That is true. Let's see what's happening on YouTube.
Ma'am, there are no questions as of now in YouTube.
True, there aren't.
There are no questions.
A lot of friends telling me that it was interesting. Thank you, everybody.
Yes.
There is a question.
Yes.
Dr. Sandhya ma'am, Dr. Daya.
Yes.
Dr. Selvi madam.
Yes.
Dr.
There is a
Bindu.
question.
There's a question.
How shall we decide Dr. Pushpanjali Das, how shall we decide the sample size to decide the sensitivity and specificity of our studies?
Wow, okay, that's a lot. Sample size. So essentially, sample size really depends on what you're trying to prove and what has been proven before you. So there's this whole very complicated, and I have to be very frank to tell you that I cannot really, it's something I check every time I'm about to do a study. I cannot, so it's not like I can directly tell you extempore what it is you have to have the power of the study. Now, the power of the study is decided by the measure, by what was found before. If you're having a novel study, novel idea, which no one has done before, you have a lot of leeway, frankly. So what you do then, if it's an absolutely new thing, then you can do, if you're going to do something like a student's t-test to it, because most likely you're going to be doing say a cross-sectional study. Now, what you need to do is the minimum, and what mistake everybody makes is that what is considered as minimum number you should have is a sample size of 30. It is not that is the best size to have, it's the minimum that you can have. And also what tends to depends on how many people are, in what section. Like, so for example, if you have 3 groups and all 3 are important to you, then you cannot have 10, 10, 10, and then say I have 30. That doesn't count. Within that main group, you should have 30. And if it's a very rare condition when you cannot have, say, for example, you're looking for a very rare cancer, where you're not going to have that many, then you can double your, what I would say, your control. Because finally, what is the whole study going to say is that what you found is not a matter of chance. That is what our p-value is. It's not a matter of chance. So if you are not going to get your question of interest, your sample of interest in the large enough size, you can still prove that it is not a matter of chance by including more of the normal or of the other groups. But essentially, that is the way I would like to explain it, is the numbers have to be like that. And a very good way is, if you're doing a study that is a follow-up of another study, please check what they did, find out what was the size, what they found. And almost every study will have shortcomings at the end where they will tell you our sample size was too small. So what you do then is you find out and you try and calculate from there, and a biostatistician will help. To get you a better answer, I will try and get someone on the channel who's an expert and who will explain to you in a much better way how to come up with the power of a study and work out on that. Aman may have some answer. Aman, anything?
I think it depends. Usually, when we are proceeding with it, like a population's, the sample which you collect should be representative of the population should be kept in mind. Otherwise, the best way to get rid of it is to say it is a pilot attempt if it is a small sample you're aiming. Otherwise, the acceptability, the power of the study, the expected effect size, we were made to learn all those 6 tips, underlying event rate, and then I've recently actually appeared for my what is that, PhD first part. So I was made to mug up all those standard deviation in the population. That was a 6th way. So these all are the reasons which you will tell your statistician to calculate the sample size. There are formulas available. There are the minimum size also available. It is always better to have more the numbers, because the more sample size will be representing the more the population. But then there are various formulas available for calculation. Better that you involve a statistician for. Otherwise, the easiest way is that, as Madam said, above 30, call it a pilot attempt.
Yes. Anything else?
Madam, if it is a longitudinal study where you're doing a follow-up, I think, and if it's a rare disease, then you can take lesser amount, lesser sample size. But if you're doing a cross-sectional study and doing it on a very common disease, then you'll have to increase your sample size. So that is basically
yes, very true. The sample size also depends on how common is the condition, definitely. And also, like you said, but it all depends. Like for example, if you are doing a cohort, again, you need very large numbers. Because remember, the cohort is supposed to represent the population. Yes. Now, one thing that I have found helps is if you compare. Now, if you basically, if you're doing a cohort, means someone else has already done a pilot somewhere. And now you are expanding on that. Either you have done it or someone else has done it. So one of the things that you can do is you should find out what was the confidence interval of the previous study. Now, for example, if the confidence interval, if the study was involving ratios, and as far as ratios go, 1 is the number that is important. So if the confidence interval included the number 1 in its range, right? So let's say it went from 0.5 to 1.5, which means it includes the number 1. So then basically your original, that study's sample did not represent the population. So what it means now, one of two things: either the hypothesis itself was wrong, or the second thing is maybe it's some technological, issues with the process, or basically your sample size was too small, right? So it could not set off the differences. It is where ratios are concerned, if it includes the number 1, and where differences are and averages are concerned, it should not include the number 0. So you should not go from negative to positive. If you are looking at anything, you should look at the confidence interval, and that is what tells you really what could have been wrong in the original study. Because finally, any second study, the way I think of it, any research that you do, the pharma industry has this very interesting thing. They believe you should either be first in line or best in line. Otherwise, there's just no point.
Or be different, ma'am.
Yeah. So either be doing something for the first time, or then you'll be giving the best answer that was there. If you're going to just go slightly to the right and slightly to the left, then, sort of just shift within the averages that were already there and give a little bit different, I don't think it is worth doing that research. And I think it's a waste of resources, of time, when it could be spent on something more useful. So always find out the shortcomings of the previous study. Do not feel bad. Everybody's study would have shortcomings. Good authors, actually speaking, I think the best person to talk to, if you want to work on someone's work that was done previously, get in touch with that author and ask them, what could you have done better? I'm going to work on the same thing. What should I do? What should I improve? What should I increase? Because someone has spent a year and a half working and writing up something, they know a little more about what the next study. And eventually everyone who's doing research is doing it because they want it to go ahead. They want it to go one step ahead. The cumulative human learning process. So ask the previous author, what went wrong? What could you add? What would you do better? And normally they will have a lot of the answers. I think that's a place to start. And finally, no one is God. We all make mistakes and we learn from our mistakes. So there should be no problem for any good scientist to come up and say, yeah, I made this mistake. So that says you learned from it. So that's what matters. Right. So anyone else?
No, ma'am. No one else has posted.
No, nothing else. Okay.
Ma'am.
Yes.
Thing is, I had posted the link to your talk on various groups, and one of my very senior mentor has written, let me know when you end up getting a perfect research so that I can get you a Nobel Prize. What he meant to say was that there is nothing which will be perfect research ever.
No, it wasn't supposed to be the perfect research. It was the perfect topic.
I think in his own way he was correct, because there is.
Oh, we lost Dr. Aman.
Yes, ma'am. There are a lot of messages. Always absolute delight to watch it. Very intriguing. It's always learning experience, ma'am. Really loved your talk. In-depth algorithm. In fact, thank you so much.
Thank you, everyone.
I think we have lost Dr. Aman.
Yes.
Dr. Aman, you have muted yourself. Unmute.
Yeah.
Yeah.
Okay, so you were telling, yes.
Ma'am, I was telling you something.
Yes.
I had, while you were talking, I called him up. What's wrong? Why that sarcasm came? So what exactly he meant was that there will be always scope of improvement whenever you do a study, whenever you do a research. It will end up having the next person who's going to attempt is going to improve it. Is going to maybe that as the technology improves, maybe 10 years down the line. So that perfect word will be not attempted by even Madame Curie and with Newton even, because we have improved that thing also. So he made his point that, you know, I would have loved if you would have said getting a apt research rather than a perfect research.
Okay. Yeah, I guess I did not put myself across very well. The idea was not as imperfect in terms of the last word.
Exactly.
But in the fact that a perfect topic that helps you to get through the whole process.
Exactly.
In relation to the process. There will never be a final word by anybody in anything, because science is expanding. There is nothing. Yes, there's. I think the topic could be perfect for its own time, for its own day. It will not be perfect tomorrow.
In a particular situation, it is perfect.
Yes. It's just for it to run properly, you know, to run smoothly in coordination with everything. Perfect in that term. But yes, good point. I possibly should have named it better. Yeah. If it leaves questions in people's minds, then it was not clear enough.
I'm glad he responded. That's the main reason. It could open up something.
Yeah, no worries. It's good. So, yep. I guess we are good to close the session. There is nothing else happening anywhere. Yes. Thank you all. And right, I shall have to do another bit of a share. Give me a moment. Okay. Sorry, I have two different computer mice. I wonder if the plural of these mouse also is mice. I never thought about it. I mean, I would hate to see a mouse running across my desk. It would just be like, anyway. Okay, let's go. Let me do this thing. Thank you so much. And I would also like to thank Dr. Aman, who was our expert today, and all the viewers for coming and being here. Now, coming this Friday at 6 o'clock IST, please note it is Friday and not Tuesday. It is Friday, the 30th, is going to be a talk on dangerous mucosa by probably someone who's written and talked more about it than anyone else. Professor Jerry E. Boko, he is going to be talking on this. I hope you will join us. It should be very, very interesting, and I hope you will be there with us. And yes, so with that, I would like to say thank you to everybody. Please, if you are enjoying and liking this content and you haven't done it before, please hit the subscribe button. It helps. Thank you so much. Thank you for being here.
Thank you
And
so much.
bye-bye. Bye, everyone. Bye.
Thank you, ma'am. Thank you, ma'am. See you.